What does that mean exactly? This is where exchange takes place. I liked the little example your textbook gave. And I was like sitting there and I was like thinking about that example because, you know, capillaries are microscopic, which means it's very difficult for us to study them, you know, really deep into the body. So they study them based on your nail bed and your cuticles because they can actually visualize them at that level. I know, it was really cool, okay? Now on the nail bed, I was kind of like, okay, what do? Do they rip people's nails out? I don't know, just saying. Okay, so they were talking about, all right, well, we understand what happens at the capillaries based on what we studied at that level. I was just like thinking, that's pretty cool, you know, thinking about all those little capillary beds in like the cuticle around my nail. But then I was like, well, wait a minute. I regularly get that cut off. All right, you know, trim down, whatever you want to call it. But still, anyway, I know that throughout all these other tissues of my body, I've got capillary beds. I've got capillary beds in the organs, the skeleton, the muscular tissue. I have got capillary beds everywhere. What does it exactly mean for it to be an exchange vessel? This is the point that when we get to structure of the vessel, that it is one layer thick. It is a single layer of cells. When I get to my capillary bed, arteries meet the veins. Those vessels are a single layer of cells. The structure of those cells allows for movement of materials.
At that point, what type of tissue are they?
You mean like the vessels then?
Like epithelial?
Yes, they're for the most part simple squamous epithelium. Mm-hmm. Little different structure depending on certain areas. So, if we have arteries, veins, microscopic, single layer of cells, okay, and the cells that make them up, their structure allows for movement of materials. So, to give you an idea, and I think your book mentioned this too, that red blood cell that made its way out of the left ventricle, we said it takes what? About a minute before it returns back to the heart, okay? As that red blood cell is moving throughout the body, and it finally decides to quote unquote make its pit stop at this particular capillary bed, okay? When it gets to the arterial side of that capillary bed, those vessels are so small that that red blood cell is squeezing through one of the capillaries, and it actually has to get slowed down as it squeezes through and meets the venules. That is how microscopic they are, and there's a good reason for the microscopic structure. Just that little red blood cell alone that made it into that capillary bed, making it to that structure, getting squeezed through, okay, it helps it to release its oxygen.
Because of how small it is?
Well, the fact that the capillary is so small that that red blood cell gets squeezed, and by squeezing that red blood cell, it helps to squeeze off oxygen. That's pretty cool. So, this business end, where we've got arteries meeting veins, okay? Arteries are bringing materials in. Veins are picking up waste to be removed. That's their job. This is the point that we maintain the tissues of the body. It's not in the big vessels. It is this point, this microvasculature. Have y'all seen all the doctors and everything that are into microvascular surgery, that sort of thing? This is a huge area now, okay? Because they are working on blood supply, restoring it, for example, to certain areas of the body. Now, the types of these capillary beds, okay, these ones that exist, for the most part, okay, the structure is going to be, for the vessels, is going to be considered continuous. For the most part, we're going to have arteries meet veins, okay? Stuff will get dropped off, stuff will get picked up, get returned, blah, blah, blah. There are cells that exist in the vessels at these levels. The ones that are continuous, cells are held together by the tight junctions, but they're going to have what we term intercellular clefts. Now, if we think back to our structure of cells, and if I've got just a single layer, okay, I'm going to have some tight junctions that exist. But at this level, I'm going to also have a little cleft that exists. So, for my tube, my vessel, okay, at this point, it's going to be a single layer of cells, and the cells are kind of being held together with those little junctions, but at certain points, there will be little breaks, little clefts that exist. And because that's there, and it's only a single layer of cells, materials can move out of the blood or back into it. Since we're talking about capillaries at this point for the arteries, okay, it would be moving materials out. So the materials would go from the blood, if this is my blood, on this side, And if these are my tissues on this side, the material would move into my tissues, into that extracellular fluid. Do you guys remember that? Okay, so I would have materials cross, be in this extracellular fluid, and let's say this cell right here needed some of these materials that just crossed over. It would kind of call out to those materials, and those materials could move into the cell. For example, if that were glucose, okay? If glucose moved and the cell needed it, it would go into the cell to be used for cellular respiration. Does that make sense? So it's at the capillaries the business occurs. Materials will move out of the blood into the extracellular fluid for the cells to use it. Now, we also find that we're going to find fenestrated capillaries. When they are fenestrated, they have a filtration pore. Okay, so think about having my layer of cells for the blood vessel, okay? But then there's this great big huge pore that's present. If this is the area of my blood, okay, that means my nutrients are in the blood, and this pore is now bigger than what I found with a cleft. So that means a substance that's bigger can now cross, or I can have rapid movement of smaller particles. Over here, it may have been that only 3 or 4 got to move through. But over here, with the huge pore, I might have 10 glucoses that can get across at one time, rather than 3 or 4. So in the structure now, it's a little bit bigger of a gap.
And they're between the tight junctions.
between the cells. Yeah, between the cells. So I get rapid passage of materials. Now, this is good for organs that have to have rapid movement. The best examples I can give you: kidneys. If we look at the structural relationship of the kidneys to the heart, this is the first, well, technically the second, but on the descent. On the descent, okay, the descending aorta, this is the first organ they hit. So the blood from the heart, as it descends down the aorta, the first organ it hits is the kidneys. The job of the kidneys, filter the blood. So there's a huge amount of blood every heartbeat that is rushing to the kidneys And because it has that structure, the materials that would be considered waste would flow rapidly out of the blood so the kidneys could filter.
Doesn't the liver do something too, though?
Yes, but the liver has even bigger spaces. The liver has sinusoids. So in my structure, in the liver, a great big, huge gap exists.
So does that mean that the liver is breaking down larger materials then? Is that
It could be, yes, because the liver needs to be able to break down. For example, in these other ones right here, we do not want an actual, for example, an actual red blood cell to pass. We don't want a protein to pass. But the liver has the ability to do that. So we would want the movement of a big particle, like a protein, a red blood cell, to pass for the liver to break it down. That's why it would be such a huge gap. It would actually allow, for example, red blood cells to move and be destroyed by the liver. It would allow proteins to cross over and be destroyed by the liver. But the other ones, no, we don't want that. We want what's in, we only want the actual nutrients that are present.
Do you find capillaries in you find it in every organ then, I'm assuming, right?
Yes.
And though in every
Everywhere.
Okay.
Yeah, everywhere. Can't even begin to count. Wouldn't want to have to do that either. So that capillary bed, the actual area where the artery meets the veins, it is this web-like structure that exists. The flow of the blood through that capillary bed is usually regulated by the arterioles upstream to it. So if the capillary bed next to it didn't get any blood, then it may or may not get blood. So usually, if it's trying to bypass that area of the body, if the one next to it didn't get blood, that one's not going to get blood, this one's not going to get blood, this one's not going to get blood, blood's going to move and go somewhere else. If those tissues need the blood supply, that capillary bed gets it, chances are the next one's going to get it, the next one's going to get it, the next one's going to get it, until it says, hey, I'm good, go on by this time. So usually, the control is from the arterioles before ahead, upstream. However, if met arterioles are present, they're going to have smooth muscle. So on that artery side, if it's a met arteriole, they have this ring, this sphincter, this ring of smooth muscle. What innervates smooth muscle? Nerve fibers. From what part of the nervous system?
No, it's
the autonomic. So think about your parasympathetic, sympathetic, that sort of thing. And what it'll do, that artery, that little arteriole that has the smooth muscle, because it's a sphincter muscle, sphincters do this number. They constrict and they relax. They constrict and they relax. So if that sphincter muscle constricted, blood flow would cut off to that capillary bed. If it dilated, blood flow would return. It's another way to control the flow to those capillary beds. So, capillary beds, the business end, if I were to cut off the blood flow to that capillary bed, I'm cutting off the delivery of the nutrients to that tissue. And I may or may not be picking up any waste on the vein, the venule side. Does that make sense? All right. So, veins. Oh, gotta love veins. With veins, these are our afferent vessels of the cardiovascular system. Afferent means which direction? Towards. So we now have to have the blood that left the heart, made it to the capillary beds. We now have the artery meeting the vein for the return back to the heart. This time to the right side of the heart. It's going to return by way of the inferior and superior vena cava. Veins throughout our body are also known as capacitance vessels. Capacity. Because of their structure. Their structure allows them to accommodate an increased amount of blood. Good or bad?
It's good if they can accommodate. If they didn't, then it hurts.
It's both good and bad. Under normal conditions, it's good. And their structure does allow them to expand. Problem is, we don't want them expanding a lot. Okay? All right? Like with varicose veins and stuff. But anyway, they have the ability to accommodate, hold larger amounts of blood than the arteries. So take a break, back at 9:30.